Sincere thanks, Adam, for the invitation. I'll admit to you that I was kind of wondering today if I'd hit the mark and I'd got the messaging right. And after hearing Ken's introduction and Adam's presentation, I hope I have. I feel relieved and validated that our approach is on the right path. And yeah, let me tell you about some of the things that we've been building over in Sydney, Australia. So I'm sure many of you have heard about Zero from various colleagues. Geez, I am tired, gosh. All right, so the Zero Childhood Cancer Program is our national comprehensive precision medicine program, been running since about 2015. Many of you will have seen this slide before. We're nearing the end of the PRISM trial, which is focused on children with high-risk disease, any high-risk cancer or rare cancers. And we performed comprehensive molecular profiling, high-throughput drug screening, PDX modeling on every patient. There are about 1,000 patients under the age of 21 with childhood cancer. And at any one time, about 2,000 patients being treated at 9 different hospitals. So I'm very pleased to tell you today about 02, which is the next phase, the next clinical trial in the Zero program. That'll take us through till 2025. And I'm very pleased to tell you that all children newly diagnosed with low risk and high risk cancer will be eligible for enrolling on this trial. And each patient will then be able to undertake very comprehensive molecular profiling. Today, I'm not going to talk about the science. I'm really going to talk about the engineering. And so it's really validating to hear the introduction again, because sometimes to be able to do precision medicine well at the national scale, you've got to get the engineering right. And that's what I'm going to try and tell you about today. So, right. So 02, it's going to open this quarter, maybe even this month. But essentially, if you're familiar with what Zero was doing already, the PRISM trial, we're doing the same comprehensive molecular profiling for each patient. That's about 100x depth tumor, whole genome sequencing, 30 to 40x germline, whole genome, RNA sequencing and methylome profiling. As before, all high risk and rare cancers are eligible, and the focus there is still on getting the diagnosis right and finding treatments for those patients. Where possible, we'll also do high throughput drug screening and PDX modeling if there's enough biomaterials to do so. And what's different is that if the patient relapses or progresses, we'll do repeat biopsy and repeat sequencing again. What's really new though, is that all non-high risk cancers will be eligible. And so here the focus is less on treatment, more on getting the diagnosis and the prognosis right as quickly as we can, and get that molecular information into the pathologist's hands who need it often to help subtype and get the diagnosis right for the patients. We can't open this to all patients immediately, so we're doing this in disease streams, and we'll open with all CNS patients first. And at a certain time after, we'll open the next disease stream, maybe neuroblastoma, maybe sarcoma, we're not sure yet. The way we're going to try and handle the patient numbers is to restrict the analysis to virtual gene panels created in consultation with our disease experts around the country and validated using data from CBioPortal, PedCBioPortal, PCAN, and our own data. And our goal here is to actually do this with a semi-automated molecular report with a 3-week turnaround. There'll be 9 sites around Australia, and we're opening 2 in New Zealand as well, opening soon. And one of the main things we're trying to do, this is not a treatment trial, it's a molecular profiling trial. And so we stream those molecular results to other treatment trials so that the patients can get targeted therapies if appropriate. And we have a basket trial opening. And there's also comprehensive germline trios and e-consent and a whole bunch of other studies going on. So I put this in because I'm pretty sure Adam's smiling right now, is that we're huge fans of using whole genome sequencing to profile patient tumors. I believe sincerely that this is an incredible, powerful tool for diagnostics and discovery. You can find all types of structural variations, single nucleotide changes. I'm going to be asleep, so I'm going to miss the extra chromosomal DNA talk, but yes, I can totally echo the use of this data to discover those things and other complex structural variations. So this is a really powerful tool for doing precision medicine at scale. It's becoming simpler through developing new methods, and it's becoming cheaper. So really that's about it for discovery. What I want to talk to you about is 02, and I'm a computational biologist, and so I see this as being a huge data challenge. Here, over every quarter, we're going to start to see new patients enrolling and rapidly climbing up to about 4,000 patients. This is all types of cancer, about 40% might be brain tumors. So pretty big numbers for us. The data ramps up fast, up to 2.5 petabytes quite quickly. And by the time we're running in full steam, we're doing 1.5 years of compute per day. And so obviously we need some pretty impressive computing resources that Ken's already touched on that are available at Amazon. So we've had to scale up my team. We're now at 16 to try and meet this challenge. I'm more a biologist, to be honest, more of a bioinformatician that develops tools to understand genome structure. But we really had to grow the team in software engineering, data engineering to build those robust pipelines, and of course data science, so we can begin to make insights into all of that data that we're generating. Unlike most of my colleagues at Children's Cancer Institute, we partner with commercial organizations all the time. You can see some of the logos down the bottom there. So I think I'm validated by this approach from what I've heard this morning already. What have we been doing for the last couple of years while we have been trying to meet the challenge of scaling up this program? We've developed a lot of computational infrastructure. Everything in blue here I'm going to attempt to cover in a bit more detail. So we focused on building computational infrastructure, new analysis pipelines, new analytics, new tools to identify interesting parts of the genome and the transcriptome that are medically important, new curation tools to help interpret variants better, a new knowledge base, new tools to help curate that information, new tools to help report it back to the referring clinician fast, and new tools to try and share that data with the research community. So again, lots of things that Adam has already talked about that are being built by CBTN and the Kids First Data Resource Center. I would say we're a couple of years behind you all in that regard, but there's lots of similarities here in our approach. So if you've ever flown into Sydney, you will have landed near Mascot, which is on the coast, and that's exactly where the internet cables come in. And so we've moved our data center into Equinix. Many of you will know that's a huge data center network, the largest, I think, data center company in the world. And so our servers are right in the same data center as Amazon and Microsoft in Sydney. So that gives us super fast connectivity to the cloud. And so this is called hybrid cloud technology. We partnered with NetApp so that we can store all of our data on their essentially file server, and we can expose that data using the types of protocols that you use on the cloud, things like S3. So that was a big win. I don't know anyone else in Australia that's using hybrid cloud. Last time I presented was back in May last year. Thanks again for that invitation. And I talked about the Biocommons Pathfinder project. And the one slide summary of that was my first collaboration with Alison and Adam and Jack from Seven Bridges. Oh, geez. I think I've got an old recording there. The take-home message from the last slide, if I flick back in a second, is that we've quartered our costs and got the analysis time down to about 30 to 24 hours to analyze the whole genome data by switching to the Cowley platform that you've already heard about today. Perhaps the biggest unexpected win from doing this is not just reducing the cost, which was a huge relief when we're doing 8 times more patients, but using that hybrid cloud architecture, we can mount our data straight into the analysis platform, which makes it heaps easier to get data in and out, which is a huge win for us. To take advantage of Cowley, we had to re-engineer all of our analysis pipelines. And so we built a thing called Graphene. It's our workhorse for analyzing whole genome sequencing data, similar to the one that Adam showed earlier, where we can just bolt in all these different analysis modules. Again, that's quartered our costs, and we can squash about 5,000 hours of compute down into 24 actual hours to get the job done. The next bottleneck, though, as many of you will know, is how you interpret and curate each variant found in the patients. How do we present that information in a molecular tumor board, and how do we put that on a report? This is the major bottleneck. We just did some health economic studies, and it takes the patient 7 weeks from enrollment The current hands-on time is about 22 hours by the time you curate the case, create the PowerPoint presentations for MTB and report. And that costs about $2,500 Australian pesos to do that. And so our goal is, by building new tools, to turn this down to a 3-week turnaround time for the patient, but a 70% reduction in hands-on time to actually do this thing. You're not supposed to be able to read this slide. It's just all the different things you have to consider when you're curating data from whole genome sequencing for patients. So I'm going to show you quite quickly some screenshots of a new platform that we built called Zero Dash to help curate the patient data. So here you land on your dashboard and you see all the patients that need curating. Here's just a couple of patients at the outset. We rapidly move into the quality control check. We can see that the data, people in my data team have passed the data. This is an osteosarcoma, so it's got a heavily rearranged complex cancer genome with p53 mutation. So already we're getting insights into the biology of this tumor just from looking at the Circos plot. You can rapidly move over to curate the somatic single nucleotide variants. I don't have time to tell you about our helium score, but it uses machine learning to try and rank the variants that are most likely of interest at the top of the list. And so really there's only two variants here worth having a look at. This p53 is pathogenic. This little icon tells us it was also found in the germline, and we lost the wild type allele in the tumor. You can see how we put careful curated notes behind each variant to help validate the curation of that particular genetic change. Here's a quick example of structural variant. Here we've got a disruption to ATRX that the team has deemed to be pathogenic as well. I don't have time to do that complicated diagram any justice, but we can zoom in and start to get some insights into the genomic architecture of these complex structural variations that are often damaging these oncogenes. Cytogenetics. And really the piece de resistance is we've now built a semi-automated molecular reporting module so that we can get from variants to report really rapidly and really reduce that bottleneck, because at the moment, pediatric precision oncology fellows are doing that type of reporting for us. So it's occurred to us that we've reported over 2,200 variants, and every time a scientist and a clinician has written a paragraph of text about that information, and we've now got enough information that we've basically can build a knowledge base. So here's what happens when you look at p53 in our data. You can see the distribution of mutations throughout our cohort, all the different cancer types. We've embedded the PCAN browser, the protein paint browser, sorry, from the St. Jude team. This is actually our data using their visualization tool. So you can start to see the hotspot mutations really clearly here. You can start to see what we actually did every time we saw all of these different variants. Did we say they were targetable? Did we leave a comment in the molecular tumor board report? And you can start to see that text. So at one point, you can either just reuse the text to write your report faster, or we can start to think about using natural language processing, turning this information into knowledge graphs and using this to help write these reports in the future, something that Ken sort of touched on, I think. So what's next in the last couple of minutes? I added this slide because of Ken's talk. This is the first time I've presented this type of slide. But essentially, I think we've got the components of building a data lakehouse. We've got all of the genomic data stored in a genomic database, soon to be about 4,000 patients. We've got drug screening data in a preclinical database along with drugs. We've got all of those curated variants that I've told you about. We've got clinical data that's a whole separate talk on how we can analyze that, biospecimens, all this knowledge that I've told you about. And we've got all the data in this net app. So I think that's the next one of the projects that's coming up next year is to work out what a data lake can do for precision medicine. Do we need it? What problems does it solve? And so I think that validates our approach a bit to hear what Ken had to say. We've just been funded my group's been funded for 4 years, where I'm co-leading the Luminesce Alliance data enabling platform, because the rare disease community is clamoring for these genome analytic techniques as well. So slightly expanding the scope of our practice. But what's really exciting to me and why I'm telling you today is that I'm now partnering with with Natasha, who's an expert in data linkage, and Tom, who's a clinician, who's one of the EMR data experts, so we can try and extract data out of the EMR, as good or bad as that might be, to try and reduce the bottleneck of data capture at the hospital interface. And I think just rapidly, because of how good the Kids First Data Resource Consortium portal is, I really wanted to leave you with the message that we're thinking very carefully about how we share genomic data. I think Adam's kind of shown you that you can imagine writing these types of queries where you can find all patients with a particular diagnosis of a particular age with a particular mutation who had a particular survival after their diagnosis. It's quite difficult to bring all this information together, but if we can get the infrastructure right, you might be able to find data that matches these types of queries furthermore across many different participating sites across Australia. So we partnered with the Australian Biocommons to try and achieve this. We're using the Beacon V2 API from Global Alliance to try and achieve this, but I don't have much time to give you any of the details. Yeah, we've been tracking all the files in the system, just like Adam has shown you, behind the scenes, so we can track all the BAM files linked to each patient. And when we add on a few more authentication frameworks, I think we can make this work. So the last piece really made me smile, because you just saw it from Adam in the new feature of PADC Bioportal. This really this is interactive, but this is in a screenshot, so it's not interactive right now. But really, how do we overlay all the information that's carefully collected, each of these from an MRI or a CT scan that captures that the patient had a diagnosis, a relapse, partial response, stable disease. I haven't even told you about the liquid biopsy initiatives that we're starting, but to truly track tumor burden over time, how that changes, what drugs the patient got. This blue drug they got because of the precision medicine intervention, so it's a targeted therapy. We can see patient Karnofsky scores declining. We see the biospecimens down here and what we actually did to interpret their data. So I think it's really cool. This is just a really starting point to try and do this. But yeah, again, validating. So huge thanks to my team and the funders that fund my team. Huge thanks to the Huge Zero team, because we've got to be over 80 people participating in this site, and all the clinicians around the country. Of course, all the patients and the families for their trust that they give us in enrolling in the study, and a huge number of people who support the Zero Childhood Cancer Program. So I think the final message really is you've got to get the engineering right to do the precision medicine at scale. And that's hopefully what I've tried to convince you is what we're trying to do over in Australia. So thank you very much again for the invite. And I hope my 1:30 ramble was audible, and happily take any questions.